Today's lab, we're going to talk a little bit about a tool called the microscope. The microscope was invented in the early 1800s by a gentleman named Leeuwenhoek, who was a lens grinder. And what he did was develop an instrument that allowed him to look at structures smaller than the naked eye could see. So basically, it magnified very tiny things. And one of the first things that he looked at was a slide of cork, which, when he observed it under his early microscope, had little air pockets in it, which he called cells. Okay? This caused him to look at other living material, and he found that every piece of living material he looked at had a cellular structure. Okay? And this was one of the big clues into the building blocks of life. This is how we were able to visualize what we're made of for the first time. Okay? And this formed one of the important aspects of what's known as the cell theory, which is that all cells come from pre-existing cells and all living things are made of cells. Okay, before the observation under the microscope, we had no idea. Okay, and because of this, it often led to a lot of misconceptions about the nature of the world around us, because humans instinctively only believe what they can see. So frequently, if we take examples from human history before the invention of the microscope, right? We used to believe that in some societies that disease was caused by demons living in your brain, and so the ancient Mayans would basically take a hammer and a chisel to the affected individual and cut a hole in their head to let the demons out. And you might think that this would have a 100% fatality rate, but actually a lot of those individuals survived because there's evidence that the wounds healed over in many cases. But this shows essentially a lack of insight into the working of the human machine. In the Middle Ages, okay, the job of barber and dentist and surgeon and doctor were all held by the same person, okay? And they believed that your health was determined by the balance of fluids in your body: blood, phlegm, sweat, and, uh, and, and bile, okay, were the big fluids that they would try to balance. And so when you would go to this, he's basically the barber, right? He would bleed you in order to fix you. So he would apply leeches, or he would cut you, and he would remove some of your blood. And then after a certain amount of blood was let out, he would pronounce you as cured. Okay, and then he would dry his bloody rags on a white pole outside his door, and believe it or not, that's the origin of the barbershop pole. Okay, um, with the invention of the microscope, we were able to have insight into a lot of these processes for the first time because we were able to see them for the first time. This led to advances such as the cell theory, which we already talked about, and the germ theory of disease, uh, which basically states that infectious disease, can be caused by organisms smaller than can be seen with the human eye. Okay, it also led to a theory in biology, in microbiology, called Koch's postulates, which state that um, if you take infected tissue and you look at it under the microscope and identify a particular microbe there by staining for it, and then you take that microbe and you grow it up in a nutrient culture, and then isolate from that culture that microbe, and then infect healthy tissue in another organism and generate the same symptoms, and then are able to re-isolate that organism from that newly infected organism, then that's probably the microbe that's causing the disease. Okay, um, and again, without the microscope, none of this would be possible. Okay, so there's different kinds of microscopes. There are light microscopes, which are the kind that we're going to talk about today and that we're going to simulate on the website. There are electron microscopes which use beams of electrons instead of visible light in order to see the specimen. And there are fluorescence microscopes which use lasers in order to visualize the specimen. Okay? Average cost of a light microscope between 500 and 1,000 bucks. Average cost of an electron microscope between $500,000 to $1 million. So you usually get one of those per institution. Okay? And a fluorescence microscope, you might be talking $250,000, okay, on average. So the reason for the difference in price is because of the cost of the components of the instrument that use electrons in the case of the electron microscope to see the specimen, and use lasers in the case of the fluorescence microscope to visualize the specimen. It costs more, okay? The reason people use these more expensive microscopes is because they can see detail at greater magnification, right? They're able to pick up even tinier stuff than a light microscope could, because they use something that has a smaller wavelength, and as a result, the tiny wavelength means that it can bounce off a tiny object and create an image, okay? Whereas something with a big wavelength will slip right by a tiny object and not be deflected and won't create an image. Okay? So the type of light source, so to speak, determines how tiny the thing is that you can visualize. Okay? The purpose of the lasers in a fluorescence microscope is to hit dyes that are attached to probes that are attached to the specimen on the slide, and that causes them to glow a particular color, and that allows us to locate things like particular proteins or particular messenger RNAs or other cellular components, and see where they are in a preserved sample. Okay? So that allows us insight into how the cell works at what we would call the microscopic or the cellular level. Okay? So different strokes for different folks. So this is a little discussion about the use of the light microscope. So you'll want to listen.
Hi, I'm Joelle, and I'm going to show you a few helpful hints on how to use the compound microscope. These pointers will help you use the microscope successfully. Before we begin, there are a few things to take notice of on your microscope. First, you want to make sure that the condenser knob is turned all the way up. Be sure not to confuse this knob with the focus knobs. You want to make sure that your fine focus is in mid-range. You can do this by lining up the white line with the edge of the coarse focus. Next, you want to make sure that your oculars are even. You can do this by setting your right ocular at 64 and your left ocular at 64. Next, you're going to make sure that your blue filter is in place. Lastly, you want to make sure that the 4x objective is in place. Now we are ready to turn on the microscope and begin. Turn on your microscope and raise the rheostat all the way to 10. Now we are ready to put the slide on the stage. For this demonstration, we're going to be using the cross-section of a human artery. Make sure that your slide is not full of fingerprints. If it is, you're going to take a Kim wipe and clean it off. Pull the stage clip open. Place the slide on the stage so that it fits into the corner of the slide holder. Release the stage clip so that the slide is held securely. You can then move the slide using the XY controls. Next, we're going to move the slide so that the specimen is centered in the path of light coming through the microscope. Open the iris and move your XY controls so that the specimen is centered in the light beam. Close the iris again so that when you look through the oculars, you do not blind yourself. Before looking through the oculars, you want to raise your stage all the way up. You would do this by turning the coarse focus knob away from you. You'll notice that the stage stops moving, but the coarse focus knob continues to turn. You are now ready to view the specimen through the oculars. You need to fit the oculars to your face, and you can do this by moving them in and out. This is difficult and may take some practice, so you may want to start by setting them at the 64 mark. As you adjust the oculars, you may see some funny effects. If your eyes are too close or too far away, you may see some circles jumping around. Keeping your eyes approximately 3/4 of an inch away from the oculars works best. Just think of it as looking into the distance into a pair of binoculars. Now, slowly move the coarse focus towards you until the specimen pops into view. You will also use your fine focus to sharpen the image. Once you have done this, you can use your XY controls to move the specimen around. What you see here is the cross-section of the human artery. The empty center is where the blood would flow. The pink section is the smooth muscle, and the purple strands are elastin. These are the effects of using the coarse focus, fine focus, and XY controls. There is one important thing that you must do before going to the next objective. You must place a desired part of the specimen in the center of your field of view. We can now move to the 10x objective. In order to see the specimen clearly, you may need to increase the light by opening the iris and using the fine focus to get a sharper image. Our next step is to view the specimen under 40x. Once again, you may need to make adjustments with the light by opening the iris and using the fine focus to get a sharper image. Going to the highest objective, also known as oil immersion, takes a few more steps. You don't need to use oil immersion with every specimen that you view, but we will go through it together. To place a drop of oil on the slide, you're going to move the objectives so that the specimen is centered between the 100 and 40x objective. You can then place one drop of oil right over the specimen. We can now turn to our oil immersion lens. You may want to adjust the light and fine focus as done previously. After viewing the specimen, you're going to want to properly clean the lens and the slide. To clean the slide, you must lower your stage and remove the slide. Then you will clean it with a Kim wipe. To clean your objective, you're going to turn the objective so it's facing out and clean it with a piece of lens paper. You have now learned how to use the compound microscope properly and successfully. It's now time to shut down the microscope, but before doing so, there are a few things you need to do to ensure the success of the next user. First, you're going to make sure that your 4x objective is in place. You're going to close your iris. Then you want to turn down your rheostat all the way back down to 1, and then turn off your microscope. That's it. Good luck in lab.
We're going to use a virtual microscope in order to simulate working with a real microscope, and the website is shown in red on this slide. There are exercises on the website that you can perform in order to learn about the different microscope controls and how to utilize them, and You have a set of slides that you can choose from and focus on different aspects of in order to understand how to manipulate the slide and find interesting places to look at on a particular sample. Okay, so you'll want to go to this virtual microscope website in order to practice using a microscope. It's free. Okay, um, if we look at a real live light microscope, is an example of a typical light microscope. We have the eyepiece or the ocular, right, which has a magnification of 10 times greater than the object. Telescope adjustment knob that can basically change um, the length of the eyepiece up here so it comes into focus for your eye. Okay, this is what's called the nose piece. All right, this is the turret. These are the objective lenses. They also have different magnifications on them to enlarge the object, and the total magnification is always the product of the magnification on the objective times the magnification on the eyepiece. Okay, we have the arm, we have the coarse adjustment and the fine adjustment knob. The coarse adjustment knob moves the stage here up and down a great distance and is only used when the scanning objective, which is the lowest magnification, is snapped into place as it is here. Okay, and the reason for that is that you don't want to use the coarse adjustment on the higher magnifications because you could ram the slide into the objective and crack it and scratch the lens, and that's expensive. Okay, the fine focus is the interior knob. Okay, and it moves the stage up and down, but it does it in tinier increments. Okay, the clip holds the slide onto the stage. Okay, the condenser here focuses the light through the slide, and the iris diaphragm basically controls the amount of light that gets through to the condenser. Okay, so the diaphragm is underneath the condenser. Okay, the stage adjustment knobs, or what we call sometimes the mechanical stage, moves the slide back and forth and left and right. Okay, by causing the clips to basically move the slide over the opening where the light comes through. Okay, and there's even rulers that are vertical and horizontal on these little clips, and you can record coordinates on them and then give that slide to somebody else, and using those coordinates they can find what you were looking at. Okay, you've got down here the rheostat. Sometimes the rheostat is on the side. Here it's on the front. Okay, and this controls how bright the light is as well. This is the lamp, right? This is where the light comes through and gets into the condenser and then through the slide, and then from there through the objective, and then up through the turret, through the nose piece, and then up to the eyepieces, the oculars, and then to your eyes. Okay, and then there's usually a power switch, which can either be on the side or sometimes it's on the front or on the back. Okay, that turns the instrument on and off. When you hold the instrument, you always want to hold it by the arm and put a hand under the base so that you don't drop it, right? Otherwise, it's an expensive mistake. And you always store it with the stage all the way down, with the scanning objective snapped in place, and with the lenses cleaned off. Okay, there's lens paper for that. That's a special kind of paper that should only touch a lens. You can clean the slides off using a Kim wipe or a napkin. Okay, and then there's usually some lens cleaner, which is a lot like Windex, that you can apply in order to help you clean it up real good so that the slide and the lenses aren't smudgy. Okay, so it's just like cleaning windows. Okay, and that, of course, right there is the power cord. Okay, the total magnification on any particular setting for the objective is always the product of the magnification of the objective and the magnification of the eyepiece. Okay, so on the scanning objective, 4 times 10 gives you 40 times, so that means that the image you're looking at on scanning is 40 times bigger than the real object. On the low power or 10x objective, you see the yellow stripe there, it's 10 times 10, which is 100 times bigger. And then on the 40x objective, which is blue stripe, 40 times 10 gives you 400 times. Okay, So as the magnification on the objective goes up, the total magnification goes up, and the working distance, which is the space between the tip of the objective and the surface of the slide, goes down. Okay, the working distance is just basically the length between the tip of the objective and the surface of the slide that allows you to see the object in focus. Okay. Um, there's two properties to the use of a microscope. One is called resolution and one is called magnification. All right. Magnification is just an increase in the size of the object that you're looking at. Okay. Resolution is the ability to pick apart separate objects at different magnifications. Okay. Resolution is limited by the light source. Okay. The resolution of visible light is much less than the resolution of a beam of electrons or a laser. Okay. And magnification is basically a product of the objective and the ocular lenses that you're using. Okay. This is just a sort of a rundown of how you properly use the microscope. It was sort of covered in the video. Okay. And the reason for each of these steps is to make sure, A, that the microscope isn't abused. Okay. For instance, the lamp in the microscope is it costs about $200, so it's not cheap. So you don't want to stress the lamp too much, and that's one of the reasons why before you put the scope away or before you turn the scope on, you always want to turn the rheostat all the way down, okay, and then turn the power off, right? So that when you plug it in for the first time, you don't pop the lamp on immediately and start to burn the filament too early, okay? The other steps are designed not to scratch the lens, okay? And obviously when you carry the microscope, we want to make sure that you don't drop it. That's why we have you hold the arm and put your hand under the base when you move it from place to place, okay? One of the classic experiments you can do with a microscope is the use of a slide with a letter E on it, okay? The idea behind this slide is that on the slide itself, the letter E will look as it does here, upside down and backwards, but when you put that slide on the microscope and look at it through the oculars, what will happen is it'll be right side up, and it'll be flipped back the way it's supposed to be. And so this is meant to indicate to you that in microscope land, everything is upside down and backwards. Okay, so as you move the slide away from you physically, the image through the ocular will move towards you. As you move the slide to the left, the image under the oculars will move to the right. Okay, so that's just something you want to get used to. As you increase the magnification, the total magnification, by turning the objectives to higher and higher power, the amount of the specimen that you're looking at decreases, so the field of view goes down. That means that the image gets dimmer because there's less light coming through to hit your eyes, so you usually have to crank the light up a bit, okay? And it also means that your working distance is much less, okay? And it also allows you a way to kind of gauge the size of objects, okay? You can, using information that's available on the internet, know that the diameter of a scanning field is 2 millimeters, okay? That would be from one side of the circular field of view to the other, okay? And there's a formula that says the magnification, the total magnification times the field diameter is equal to the total magnification that you've chosen times the field diameter, okay? So if you know that on scanning the field diameter is 2 millimeters, okay, and the total magnification is 40, those are fixed values, okay, you can set that equal to the new field diameter, right, by dividing that product by the new total magnification, which you can determine by looking at the magnification on the ocular and the objective. And that'll give you your new field diameter, and it's going to be less than the scanning diameter, okay? And how much less is going to be dependent on the objective that you choose, okay? For instance, if you were to choose the 40x objective, the field of view would be 10 times less than on the scanning objective, okay? So the diameter of the field of view on the 40x objective, the high power, is 0.2 millimeters, whereas on scanning it's 2 millimeters, okay? And the reason that we do this is so that you can get a approximate idea of how big an object is, how wide it is, okay? Another traditional experiment is called depth of focus, okay, or depth of field. Here you take a slide with crossed threads on it, and they are at different depths on the slide, and what you do is you try to determine which thread is on top, and the way you do that is by, again, starting with the stage all the way down, and then you bring the stage up slowly with the coarse focus until you see a blurry image, and then you can flip to a higher power objective. Next higher power is fine, low power say, and then use the fine focus to bring the first thread into focus, and that'll be the thread that's on the top. The thread that's underneath will be the next thread that comes into focus, and the thread on the bottom will be the last one. Why do we care? Because you need to get the idea that when you work with slides, sometimes you need to focus near the top of the specimen and sometimes near the bottom of the specimen in order to see different features, okay? As magnification goes up, the depth of field goes down, okay? And as the magnification goes up, the width of the field goes down, okay? And basically, the reason for this is that you're looking at a tinier region of the specimen when the magnification goes up. Another thing you're going to learn about microscopes is that generally, when you're in focus on scanning, when you go to the next higher power, you don't need to drop the stage down or do anything. All you do is flip the next objective into place, and the image should basically be in focus and centered the same way it was on scanning, and the only adjustment you should need to make is with a little bit of the fine focus, okay? And that's a property of the microscope. It's called parfocality. A microscope that's been properly maintained will have that capability, okay? On the website, you can look at cheek cells, right? Normally in lab, if we were doing this live, you would scrape the inside of your cheek with a toothpick, and you would agitate that toothpick into a salt solution dropped onto a glass slide, which looks like a little rectangular glass window, tiny glass window, okay? And then you would stain that with a stain called methylene blue, and then you would put a little glass plate called a cover slip over the top, squish it down, blot away any extra liquid, and then you would look around for your own cheek cells, okay? So this is called a wet mount. This is what it looks like on scanning, okay? This is what it looks like on low power. That dark spot in the middle there is the nucleus of the cell where the genetic material, DNA, is contained. And that outer boundary is called the cell membrane, okay? And then the sort of light purple region between the cell membrane and the nucleus, that's called the cytoplasm, okay? These cells are simple squamous or stratified squamous epithelial cells from your cheek. This is, for instance, how they do DNA testing on people that want to know who the parent of their baby is, okay? And that's also how 23andMe does genetic testing on you if you're curious about your family's origins. They take the DNA from cheek cells and isolate it, and then they sequence it. They get the sequence of DNA building blocks, the nucleotides, and they compare it to known sequences that they get from a big database, and they can determine where your DNA came from through evolution. Kind of neat, okay? Remember, when you store the microscope, to follow these steps. If you're using an actual microscope, the website will walk you through these steps as well, okay? So make sure to familiarize yourself. What will you need to know from this lab. You'll need to know the parts of the microscope and what they do. You'll need to know how to compute total magnification, okay? And you'll need to be able to determine the parts of the microscope from different views, the front, the back, the side, and so on. Okay? All right. I will see everybody in the next podcast.